After watching this video, you should be able to describe the GS signal transduction pathway starting from ligand binding all the way to protein phosphorylation. You should also be able to list the mechanisms to turn off the GS signal transduction pathway and some important examples of GS receptors and medical applications that affect the GS pathway. So let's start with ligand binding. Okay, and you can see here that we have a 7-transmembrane spanning receptor with the N terminus on the outside of the cell and the C terminus on the inside of the cell. Now, the reason why these are called G-protein coupled receptors is that they are going to interact with a guanine nucleotide binding protein. In this case, we can see here that there are 3 different subunits: an alpha subunit, a beta subunit, and a gamma subunit. And so this is a heterotrimeric complex, and you can see here that it's bound to GDP. And in this heterotrimeric state, bound to GDP, the G protein's inactive. Now what happens after the ligand binds is that there's a conformational change in the receptor and causes the affinity for this alpha subunit for GDP to go down, and its affinity for GTP to go up. And we can see here that we have GTP coming in and it kicks GDP off, and that breaks up this heterotrimeric complex. We can see we have the beta gamma subunits over here. Now they typically don't do very much. There are some important examples that they can regulate some ion channels and some different enzymes, but most of the work, most of the business is done by the alpha subunit here. And when it's bound at a GTP, it's in the active state, and its target in this case is going to be adenylyl cyclase, and that's abbreviated AC. It's a membrane-associated protein, and when GS activates adenylyl cyclase, it takes ATP and cyclizes it, releasing a phosphate, creating a phosphodiester bond, making cyclic AMP. Now, cyclic AMP has some important effects on its own. It can act on ion channels and it can affect other proteins, but the most important aspect of cyclic AMP is that it activates a kinase that's cyclic AMP dependent. Now that's indicated here. We see that we have this big protein, it's 2 regulatory subunits, abbreviated R, and 2 catalytic subunits, abbreviated C. And this collectively is protein kinase A in the inactive state. Now, what happens is you got cyclic AMP binding sites on these regulatory subunits, and when cyclic AMP binds, there's 4 binding sites, it causes the regulatory subunits to dissociate from the catalytic subunits. And what that does is it allows the catalytic subunits now to do what their main function is, which is to phosphorylate proteins. Now, that's abbreviated here as 2C. That's indicating that there's these 2 catalytic subunits. Now we have our active pKa. We can see here we've released the regulatory subunits bound to cyclic AMP. And now the target of protein kinase A is not very specific. You know, a lot of kinases, when you look in the beginning of the name of the kinase, tells you about what they're going to do. So in this case, since it's protein kinase, it's very nonspecific. And there's a lot of targets of protein kinase A. Too many to list in this lecture, but we'll go into them in other subsequent lectures. In this case, we're going to generically just say protein, and the protein, when it's not phosphorylated, can either be more active or less active. It really depends on the protein. And in this case, we see the protein is getting phosphorylated by active pKa. And that's why we have a P indicated on the protein. And now this protein can either be activated or inactivated. It really, again, depends on the protein. There's going to be lots of examples that we'll discuss in subsequent lectures where some proteins are becoming more active and other proteins are becoming less active. In this case, the basic concept is that a protein is going to be modified and by this phosphorylation of a serine-threonine residue on the protein, and it's going to change its activity. Okay. Now, that's affecting existing proteins. Okay, so these proteins are already in the cell, and PK is going to modify their activity. Now, there's also something else interesting that happens. The catalytic subunits have a nuclear localization signal that's kind of covered up by these regulatory subunits. And when these regulatory subunits dissociate, you unmask the nuclear localization signal, and that allows this pKa molecule to actually get into the nucleus. Now, when it gets in there, what's it going to do? Well, the main thing that you ought to think about that pK does in the nucleus is it phosphorylates a protein that's already sitting in the nucleus on DNA, and that protein is CREB, C-R-E-B. And the reason why it's called CREB is that it's cyclic AMP response element binding protein. So this CREB molecule sits on a cyclic AMP response element, and when it's phosphorylated by pKa, whatever gene it's sitting on is going to modify the transcription of that protein. So not only do G proteins, in this case GS, have effects on existing proteins, but it also has genomic effects as well. And that might explain why we see some effects of these G proteins are very quick, and other effects are take a little longer to take effect. And that might be explained by the fact that you also have some genomic effects going on. All right, now that we've gone through the whole GS pathway, it's important to remember that the take-home message is that we have a ligand binding the receptor, and the net effect ultimately is we want to modify proteins. Now those proteins could be enzymes, they could be channels, they could be transporters, but the end game is that we want to change activity of proteins or even change the transcription of the protein inside the cell. Okay? And commonly, cyclic AMP is called a second messenger because the first messenger was the ligand, and now that has been handed off to cyclic AMP, which then, in this case, affects protein kinase A, which does a lot of the work of cyclic AMP. Now, we can't just have all these proteins phosphorylated now, right? We can't have the system continuously being active, otherwise we'd have a lot of problems. So we need mechanisms then to turn off the system. Okay, so this is quite important because otherwise we'd have constitutively turned on GS pathways. So the way to think about this is just to walk through all the various steps that make sense where you might want to have an effect. So for example, if we start with number 1, all right, of course, an obvious thing is that the ligand's going to unbind. These ligands that bind to the receptor on the extracellular side aren't bound irreversibly. They bind and they unbind. And so when the ligand unbinds, the conformational change will go back, and that will help turn the system off. So that kind of makes sense. That's usually true for most receptor-ligand interactions. Now another mechanism, if you think the next step in the pathway, is that the G-alpha part of the heterotrimeric complex actually has GTPase activity. So there's some intrinsic G-alpha GTPase activity. What that means is it's going to hydrolyze the GTP back to GDP. Now remember, when a GDP is bound to the alpha subunit, it reforms that heterotrimeric complex, which is now going to be in the inactive state. So this GTPase activity is actually very important, and it's intrinsic to the alpha subunit. And actually, there's some proteins inside the cell that can regulate the G-alpha GTPase activity, and that might make for an interesting drug target potentially affecting the pathway. Now, another mechanism that we can talk about that would turn off the system would be cyclic AMP phosphodiesterase activity. Okay, so we have our cyclic AMP molecule that we form from ATP, and remember it has that phosphodiester bond, and there are enzymes that break up that phosphodiester bond, and they're called phosphodiesterases. All right, so we certainly could turn off the system that way. So we have phosphodiesterase. and this is abbreviated PDE, okay? And this causes the cyclic AMP to go to AMP, and so you remove the cyclic AMP that's being produced by the cell, you don't have cyclic AMP to activate pKa, and that helps also turn the system off. Another mechanism is you can dephosphorylate the proteins that pKa phosphorylated. So there are protein phosphatases that remove the phosphate that pKa put on. All right. Now, what we could do is we can go back to our diagram, all right, and put in these different numbers. So remember we said, you know, you can have the ligand unbind, right? That was the first mechanism. We can have intrinsic GTPase activity that cleaves the GTP back to GDP and reforms that heterotrimeric complex. We could have cyclic AMP being broken down to AMP by phosphodiesterases, right? We called that number 3. And then we said that we can also have protein phosphatases that undo the work of pKa by removing the phosphate. We can call that number 4. All right, so those are very important mechanisms to turn off the pathway. Now, going back to our list, there is one other mechanism that we can talk about, and it really is something that happens when there's overstimulation of the receptor. Receptor's getting stimulated too much. And so we can call this number 5, all right? And number 5 would be that there is receptor desensitization, or downregulation. Now the way this works is that there are some enzymes and kinases inside the cell that can phosphorylate the C-terminus of the G protein, and what that does is it could decrease its activity and also attract other proteins, which ultimately can trigger internalization of the receptor and removal of receptor from the surface, making it so if there is a ligand around, it's not going to really have much to work with. And this is actually a very important mechanism that comes up all the time. There's lots of medical applications, drug applications to this that we'll talk about at the end of the video. So this is also an important way that the system can turn off as well. All right, so now that we've gone through the mechanisms of turning off the pathway, we've gone through the pathway, let's talk a little bit about some medical applications. All right, so go over here. The first thing that you should appreciate is that there are lots of receptors that are clinically relevant that are coupled to GS. All right? Comes up all the time. I'm just going to list a few that are really important. For example, there's beta adrenergic receptors, beta 1 adrenergic receptors, and beta 2 adrenergic receptors. All right? Beta 1 receptors, you find these on the heart. You find these on the cells that secrete renin, a very important part of the renin-angiotensin-aldosterone system. Beta 2 receptors you find on smooth muscle. You find on different epithelial cells that secrete aqueous humor in the eye, or even on skeletal muscle. These are very important, have very important physiological functions, and they increase cyclic AMP in their target cells. And there's drugs that either block or stimulate these receptors. It's very important, and we'll discuss these in other lectures. Other examples of GS receptors, the V2 receptor, which is the receptor for ADH, antidiuretic hormone, on the collecting duct cells in the kidney. When it activates its receptor there, it increases cyclic AMP, increases pKa, and that helps insert aquaporin water channels to help with water reabsorption in the kidney. Very important physiological function, regulated by cyclic AMP and PKA. Other examples, histamine receptors type 2, H2 receptors. You mostly find these on the parietal cell and the GI tract that help regulate the K plus H plus ATPase for acid secretion, which is very important in GI function. And it's also a target of drugs to treat gastric reflux. Blocking H2 receptors would then decrease acid secretion. Again, it's coupled to GS. The cyclic AMP helps regulate that acid secretion. So it again comes with very important medical applications here. Other examples, glucagon receptors is a very important metabolic hormone. It's released from alpha cells of the pancreas, and it helps regulate blood sugar. It's mostly what it does. And it has a lot of effects on the liver, various metabolic enzymes involved in various metabolic reactions like gluconeogenesis and glycolysis and glycogen metabolism. And it works through cyclic AMP and PKA. Okay, so this is a very important hormone in regulating blood sugar. There's other hormones. Other examples, the PTH receptor, parathyroid hormone receptor. Okay. I guess I could put R for glucagon here too. PTH receptors, they just don't only do GS, they do some other things too, but cyclic AMP and PKA is a very important part of how it has effects on calcium and phosphorus metabolism. All right. Secretin receptors. Secretin's a hormone in the GI system. It regulates bicarbonate secretion. It's very important in regulating GI function. And it's also coupled to GS, just like all these other ones. And then, you have some other receptors like GLP-1, glucagon-like protein or peptide 1. In a previous video, you may have seen the regulation of insulin secretion from the beta cell. Cyclic AMP and PK help regulate that, and the GLP-1 receptors are a very important part of modulating glucose-mediated insulin secretion. And these are drug targets, things that help increase activity at this receptor. And then you also have some eicosanoids like prostacyclin or PGI2, the PGI2 receptors that you find on vascular smooth muscle and platelets, sort of counteract platelet aggregation from thromboxane A2 and also counteract the vasoconstriction from thromboxane A2, so sort of the opposite effects that you see with thromboxane A2, an important eicosanoid released from platelets. And there's other examples like thyroid stimulating hormone, there's the gonadotropins, the list goes on and on and on. What I want you to appreciate is that there's a lot of receptors that do this, and they use cyclic AMPK to mediate their effects, and depending on what the tissue is, the effect is going to be different, and you have to really go and look at each of those receptors and the tissue to really figure out what PK is doing there. But people have mutations in these receptors that have various medical syndromes, and like I said, there's lots of drug applications, either things that block or stimulate these receptors. Comes up all the time, really important. Now, the other major medical applications would be related to things that actually are specific problems in the pathway. So one classic one is cholera toxin. All right. So cholera toxin, what it does from the Vibrio cholera bacteria is that it enters intestinal epithelial cells and it modifies the G-alpha protein in intestinal epithelial cells. And what it does is it takes an ADP ribose from an NAD molecule and it modifies the G-alpha protein in such a way where it decreases its intrinsic GTPase activity. So it decreases the G-alpha GTPase activity. Okay, now keep in mind that this is very specific for intestinal cells. So there's a receptor for the cholera toxin. It gets into the intestinal epithelial cells and it modifies specifically the G proteins there. And when you do this and you cause a massive increase in cyclic AMP, All right. In those cells, what that does is it decreases absorption of sodium from some of the intestinal cells, the villus cells, and then it also increases active chloride secretion in another group of cells in the crypts. And the net effect is you get this massive secretion of sodium chloride and massive watery diarrhea, which can be life-threatening. And so the underlying mechanism, though, is that it's causing a dramatic increase in cyclic AMP production because the G-alpha GTPase activity is not working and the system is just sort of overactive, right? It can't turn off. And so that's a really important example of that, of a problem with turning off the system. Another example is a drug application. You know, there are a class of drugs called methylxanthine drugs, okay, and these are related to caffeine or theobromine, which you find in chocolate. And these methylxanthine drugs, like theobromine or, theophylline, for example, what they do is they inhibit phosphodiesterase. In this case, in our pathway, we're blocking the cyclic AMP phosphodiesterase. It turns out a lot of these drugs are pretty non-specific. They block a lot of different types of phosphodiesterase. But the net effect, though, by blocking this is that you get a big increase in cyclic AMP. And clinically, the reason why this is helpful is that if you do this in the bronchial smooth muscle, the cyclic AMP increasing there actually relaxes the bronchial smooth muscle, and that's actually helpful for lowering airway resistance in people who have like asthma. So in this case is a therapeutic use. Okay, but again, the mechanism is that you're blocking the turning off of the pathway, so you're turning the pathway on. All right, so that's another really good example. Now finally, the fourth example that we'll discuss is that relating to the downregulation desensitization. So when you overstimulate these receptors, the cell responds by making the receptor not respond that well and even taking the receptors away. And a classic example is beta-2 agonist overuse. All right? So people need to be really careful when they're using their beta-2 agonists. You know, for example, things like albuterol, which is a short-acting beta agonist, is commonly used for people who have asthma as a rescue medication. If you take this too much, that mechanism of downregulation and desensitization kicks in, and then the drug doesn't work as well anymore. You know, and so then the person feels like they have to take more, and it just makes the problem worse, right? You downregulate and desensitize the receptors even more, and it's a vicious cycle. And so people need to be real careful not to overuse their beta-2 agonists, have some breaks, enough time in between so you don't cause this downregulation and desensitization to occur. And this isn't just specific for the beta-2 receptor. You see this with a lot of other types of receptors too, but it's a classic example. So hopefully now that we've gone through the pathway and starting with the ligand binding, activating the G protein by causing a conformational change in the receptor, shifting its affinity for the guanine nucleotides to GTP, turning on adenylate cyclase to make cyclic AMP, which then activates protein kinase A to have all these important effects on various proteins. And the mechanism to turn them off here in red, you can see there's lots of opportunities for either drugs or disease states. And so if you have a really good grasp of this GS pathway, you can understand a lot of physiology related to hormones and medications and disease. And that concludes this lecture on the G protein signal transduction pathway specifically for the GS pathway.